Chapter 18, we're going to talk about the cytoskeletal system. This whole system within a cell is very important for setting up the structure of the cell, and it also plays a large role in helping transport some of the different organelles and hold them into place. So we were talking about vesicles in earlier chapters, and those are running along some of these skeletal systems. So we're going to talk about how they provide structure, how they help to interconnect different networks in different areas of the cell, and talk about their role in cell division and movement, as well as the fact that they're very dynamic and changeable during their time in the cell. There are three different types of proteins involved in structure. There are microtubules, microfilaments, and intermediate filaments, and they're all made up of different types of proteins. We'll go through each of these, but you can see that they have different sizes, they have different organizations, and they are grouped together differently. So you can see this is kind of alpha helix of these different subunits. We have monomers that form this kind of twined, tethered rope. And then here we have the intermediate filaments, are kind of stacks of things that kind of fit together in these tight corded structures. Fluorescence microscopy is often used to visualize these different structures. So remember that we have different proteins, and we know what those proteins are made up of, so we can generate antibodies against these different types of filaments or microfilaments or microtubules, these different proteins, and then we're able to visualize those within the cell, typically using fluorescence microscopy. This is an example of actin. We have an anti-actin antibody and a red fluorophore, so that we're able to see those actin bundles within this cell. Live cell fluorescent microscopy can also be used. There are ways to use fluorescence dyes in living cells or tagged proteins. So if you know that you can tag a protein, have it be synthesized in the cell with a GFP or an RFP, so green fluorescent protein or red fluorescent protein, those fluorescent molecules can be synthesized, or sometimes they can even be micro-injected into the cell of interest. So here we can see that microtubules are shown, and I'm going to show you just the kind of dynamic nature of this. If I click on this slide here, you can kind of see the kind of look at the 145 seconds. I'm going to overlay the 0 seconds and just kind of look at that one slide. You can see how those filaments are moving after a different amount of time. So they're kind of they're dynamic in nature. So we can see how those are growing and ungrowing and kind of moving in space compared to each other. So live cell fluorescent microscopy helps us visualize those changes over time, and helps us understand that they are in fact dynamic. You can see these in high resolution images in phase contrast. So if you can computer enhanced digital photography, you can kind of use some of the lenses and the filters to kind of get a better image of these different microtubules. Here's an example of just standard microscopy, and then also there's electron microscopy. This is an image of an electron microscope can resolve individual filaments and thin sections within special mounting techniques used for electron microscopy. Microtubules we'll start with first in terms of the three different types, and they're really important for cilia and flagella structure. So big cellular movements, you can kind of see those here in this image, and they're one of the largest structural elements of the cytoskeleton. You can see that there are different components of them. So we have they're organized within these different arms of the dynein, for example. And there's a lot of different groups of these put together in order to provide the structure and the flexibility to allow these kind of like whip tails to be able to form and move. So there are two different types of microtubules for the functions in the cell. Axonemal microtubules, so they're involved in cilia and flagella, and they're organized in these different various patterns, and you don't have to memorize these or anything, but just recognize there are different ways that these can be stacked together and organized to form cilia or flagella. There are also cytoplasmic microtubules that are involved in axon formation, mitotic meiotic spindle formation during cell division, the structure of the cell, the shape, and then also vesicle movement. So these are all different. These are kind of two different types within, depending on where they are and how they're organized. So now we'll zoom in a little bit more on the microtubules and talk about what they're made up of, what kind of proteins they're made up of. So microtubules are straight, and you can see kind of this organization. They're hollow cylinders of different lengths that usually consist of about 13 arrays of polymers. They're called protofilaments. And the basic structure of a protofilament is the heterodimer. So hetero means two different types of these dimers, the alpha and beta here tubulin of these compounds. These are non-covalently attached, which means they're using intermolecular forces to form these alpha-beta heterodimers. They have really strong interactions with each other, so they don't normally dissociate very easily. So they come together and they form pretty solid structures. Here's a little video check that I'd like you to write down on a piece of paper that I'll check when we come back and meet as a group. But I want you to kind of comment on this idea. What does the thought that these are held together by non-covalent bonds, what does that infer? What does that tell us about the intermolecular forces that are keeping them together? And we'll chat about this when we meet again as a group. The interesting thing about the alpha and beta tubulin structures is that they're very similar looking, right? So if you look at this alpha beta, you can kind of see these different alpha helices here, kind of wrap around. You see these beta sheets in the middle. They look very closely identical. They look very similar to each other. But they only have about 40% of amino acid identity similarity. So even though they look structurally similar, 40% only similarity between the two in terms of their amino acid makeup. All of the dimers are oriented in the same way because of the orientation, they have this inherent polarity. So there's a plus end and a minus end that tells us where the next thing's going to attach or where it's going to break off from. So it's kind of got this orientation that's very important for when these are put together and when they're broken down. They have to be organized the same. And on this picture, there are just GDP, GTP. There are different binding sites for various different drugs or things that are shown in here that we'll talk about a little later. Microtubules can form different types of those hollow tubes. They can be singlets, doublets, triplets, for example. You can see where there's those hollow sections for these different types. So here are the protofilaments. There's 13 in each of these kind of sets, stacks, and you can see that 1, 2, or 3 sections. As I mentioned, there are GTP and GDP binding sites within that protein structure. And this is really important because GTP hydrolysis is necessary for the structure of this being held together. So if we look at this first figure here, we want to grow this microtubule. As I mentioned before, there is a directionality to this. There's a plus end and a minus end. So if we have GTP tubulin molecules add to the end of the microtubule. So we have GTP adding. So we have tubulin molecules with GTP. So it's bound in that free form, and they start to add in and cluster in here. So then once those are hydrolyzed to GDP back here, so these are GTP, GDP, then that kind of stabilizes this lagging end of the growing microtubule. And then we can add on more GTP caps. If we don't have this GTP cap, what happens if these start to fall away or we stop having GTP available in the system? That'll kind of break down, and then we won't have that cap on here anymore. If that cap's not on there, then these things start to peel away and fall off, and then we shrink the microtubule. So this is kind of the dynamic nature. This is one of the mechanisms for regulation. If we have GTP in the system, we can kind of grow these. If we have GDP or we don't have GTP in the system, then that falls off. We don't have enough to cap that, and then we start to shrink the microtubule. Microtubules can grow from both ends, the plus and minus end. Usually the plus end, you can see, obviously grows a lot faster in the presence of those monomers, and the minus end can usually be attached to other things. So this was kind of seeded with the basal body, which was some of the microtubules to begin with, and then you can see these grow off out of that in the right conditions. We also have these microtubules form by addition of tubulin dimers at their ends. So we can also, if we have this is percent tubulin in the microtubules over time, and this process is called nucleation. So the addition of more subunits at either end, and that's called elongation. So microtubule formation is slow at first because the process of nucleation is slow. So the organizing these and creating that seed for other ones to come together, that is a slow process. So this takes some time. But once it gets going enough, once we have this protofilament kind of formed, then we have that elongation phase is pretty quick and rapid. Once we get up here, we kind of lag phase, it lags out and it slows down. So we have a similar amount of these actin tubule molecules falling off and coming on simultaneously. So this is almost kind of at equilibrium here. And part of the reason is because there's, we've lost concentration of these alpha beta tubulin molecules, so we can't grow it anymore. Here's treadmilling, where we kind of, on the back end, the minus end, these start maybe falling off, and then on the front end we have growing. So we can kind of grow and move and push a cell, for example, from one area to the other, just based off of the microtubule growth. And the blue ones are just to kind of show how these are kind of cycling through from the plus as we add different microtubules on the front end. Then on the back end, those alpha beta tubulin proteins are coming off. Other proteins can stabilize or destabilize microtubules. One of those is EB1, and it regulates the positive end, the plus end of microtubules. So this protein is going to stabilize the plus end from if the GTP hydrolyzes and the cap is kind of off, it'll still kind of hold this together. So you can see here, fluorescence microscopy, EB1 staining, and microtubule, so anti-microtubulin antibodies, and then EB1 antibody. You can see that it's right just on the tips of the end caps of these on the positive end. Other proteins promote depolarization of microtubules. Staphin and OP18, they bind to the tubulin heterodimers and prevent them from further polymerization, so they keep them from growing. Catastrophins act as the ends of the microtubules, and they promote the peeling off of the end units. So some of these sever and break down these as well. There are certain drugs that also can influence microtubule strength and formation. Colchicine is one of those, and it is an inhibitor of the monomer assembly. So it promotes microtubule disassembly. It's actually used as a gout treatment, and it inhibits neutrophil activation. So autoimmune system, it slows down some of those neutrophils that would be releasing some of the inflammatory cytokines, and so it breaks down some of the those tubulins so that they can't move around as well to influence that effect, and then it can resolve some of the inflammation that's happening in gout, which is typically localized to different joints in your body, pain in typically like hands and feet. Nicotazole is another inhibitory assembly drug. It's similar to vinblastine, viniscreen, and it's a little more easily reversed than those of colchicine. You can see here that we have the green tubulin. We haven't spoken about actin yet, but this is kind of outlining the cell as well here. So this is a picture of a normal cell, a CHO cell, and here we'll show you what happens when we do the drug treatment, nicotazole. You can see that this treatment changes the structure of the cell quite substantially. The organization is disrupted, and it kind of depolarizes those microtubules, and you can see that large change in how this cell is organized before and after treatment. So here are some of the chemical agents, and you'll notice some of the chemical, the drug names, and also the sources, so where they were identified from. So these are not necessarily just acrylamide is synthetic, but a lot of these are actually coming from other plant compounds. A periwinkle plant, for example. Here's synthetic, is nicotazole is actually synthetic, but a lot of these are found in other places. Death cap fungus, phalloidin, we'll talk about that also influences microfilaments. So some of these are interesting to look at and see, and allows us to study these cellular organelles, but it also has typically a way that we can use these in certain types of treatments for various conditions. A lot of these are used in cancer treatments, and they're typically called the antimitotic drug class because they interfere with spindle assembly and then inhibit cell division. And so some of these are kind of slow down the rapid division of cancer cells and inhibits their cell duplication. Some of them, like Taxol, for example, that is used to bind up some of those. It depletes free tubulin, so it causes cell division to stop because it doesn't have tubulin to build up the structure of the cell. A few slides back, you can see this basal body, and that area is important because that microtubules typically originate from what's called the microtubule organizing center, or MTOC, and that are these little spots here shown in these various cells. If that microtubule organizing center is near the nucleus, we typically call that the centrosome. And in animal cells, the centrosome is associated with the two centrioles surrounded by pericentriolar material. So these anchor the microtubules, and then the microtubules grow out from that MTOC. Usually the negative end is more towards that centrosome or MTOC, and then the positive end moves out from there. Here's an example of a ciliated epithelial cell where you'll have that MTOC in the middle, the negative ends towards that MTOC, and then the positive ends grow out from there to both for the apical end to create these little fingers, and then on the basal end to kind of just form structure within the cell. The centrioles are made up of the microtubules, and these are in the triplet conformation. So here you can see 9 different pairs of these triplets are organized into the centriole. If we don't have centrioles, then typically the microtubules within are poorly organized. And you can see that in that last slide where we had the red blood cells. They're not necessarily, they're kind of mixed and all over the place, where they don't have those microtubules that organizing centers, then they're a little more less organized. These microtubule organizing centers or centromeres typically large protein complexes in them, and they contain this gamma tubulin. Gamma tubulin is only found in centrosomes. And here you can see this organizing center here. And these gamma tubulins, they help to nucleate and start or seed the assembly of these new microtubules away. So they kind of create this negative end and help to generate that positive end and the growth away from this organizing center. So the loss of, if we take out gamma tubulin, it prevents a cell from nucleating microtubules, and then we can't have that organized structure of the cell. These microtubule organizing centers, they have just a fixed number, a limited number of nucleation and anchoring sites. So you can kind of see where we would have, you can kind of see this out of this fluorescent image where alpha tubulins in red, DNA is in blue, and then the gamma tubulin is where we would have the organizing center is in the yellow. And you can see they can only have so many of these different spindles coming off of them here. Their function is essential for cell division, and they're involved in attaching to and pulling these different pieces of DNA apart. Other proteins that associate with microtubules, they're typically called microtubule associated proteins, or MAPs. One specific type of MAP is called tau. It is a protein that's associated with microtubule. Tau is a really important protein that actually helps to pull together microtubules to form tight bundles within axons that you can kind of see in this fluorescent image. So tau is an axonal protein, And then there's a dendritic protein that associates with bundles there called MAP2. That protein actually promotes the formation of looser bundles. And so then you have a difference between an axon, which has really tight bundles that have to grow a really long distance, and the dendrites, which typically fan out a little more and have a little looser formation of their the microtubules, and that changes the structure of the neuron and also the function of that space as well. Here's a fun little experiment using cells that normally don't have any of these types of projections that you'd see inside a neuron. These are insect cells that are just in a standard microscopy. And what happens if you overexpress the tau protein in cells that normally don't have tau, you can see they can form these long spindles. So these almost start to look like axons within this cell culture system when you overexpress this specific protein that's typically not found there. So that is the discussion on the microtubules, and next we will go into and talk about the microfilaments.